research interest is healthcare research, disease management, and translational healthcare products. His expertise ranges from biotechnology, genetics, including population genetics, evolutionary genetics, clinical and immunogenetics, and infectious diseases research. He is involved in translational healthcare research and management project. I invite you on stage, sir. As our second session chair, I would like to invite Professor Anup R. Bandyopadhyay, sir, University of Kolkata, India. Professor Anup R. Bandyopadhyay is the former head of the Department of Anthropology, University of Kolkata, and was the president of Anthropological and Behavioral Science section of the Indian Science Congress in its century year, and has published more than 100 anthropological research articles in national and international journals on evolutionary biology, forensic anthropology, dental anthropology, public health in relation to medical genetics, cytogenetics, and anthropometry of non-communicable diseases, dermatology, physics in medical and physiatric disorder, and population genetics and human rights issues. I welcome you on the stage, sir. Beginning with our first lecture, the keynote lecture by Dr. Thangaraj, sir, Director of CGFD India. His major research interest has been in the field of human origin, health, and disease. His group is interested in understanding the complex origin and affinity of Indian population using the genomic variation of contemporary population and ancient biological remains. His group is also interested in understanding the impact of endogamy on the health and disease of South Asians. His other research focus is to find molecular basis of male fertility, cardiovascular diseases, sex determination, and mitochondrial disorder. I welcome you on stage, sir.
Very good morning to all of you. Thank Dr. Chaudhary and Dr. Chandana for inviting me to talk and also organize this meeting, which is appreciated by everybody in organizing this meeting. When Janeshwar asked me to talk and he was asking about the topic which I want to speak, I choose to speak on this topic. Many people have asked the audience that where is my favorite topic of human evolution and genetic disease and so on. So I did not tell most of you, but I spoke in this very same hall on November 7th, combining my work and Thangaraj's work. Therefore, I think most of them must have heard here. Therefore, I thought I'll talk to you on different topics. Of course, this looks like a different, but there's always a combination of evolutionary genetics. When you talk about infertility, people feel whether really infertility is a problem here, because we have a population size of 141.7 crore, the increasing number of about 1% every year. People feel there's no problem of infertility. However, if you look at this particular data from UN, it's about more than 6 years data, suggests that the average number of children per woman fell down drastically. It was about 6, it has come down to 2.5 or maybe less than that now. Then again, one would think whether this reduction is really because of the infertility or there's a reason for that, whether because of government put lot of effort in controlling the population. However, I just Googled last night to look at how many infertility centers are in this city alone. There are about 18 infertility centers. This tells a story that we come across several infertility clinics all across the country. I think their problem of worldwide. The fertility rate has come down drastically. When you talk about fertility, infertility, there are about 15% of the couples are suffering from infertility, and there is no difference between male and female. Both contribute equally to this cause. I'm going to talk about only the 50% of the 15% that's male infertility. And if you look at the cost for male infertility, there are so many, including clinical costs, environmental, lifestyle, sometimes even the drug which we take to cure some disease can lead to infertility, the occupation, particularly those who use laptop all the time also found to be restricting to genetic factors. There are many chromosomal abnormalities which I talked in the coming slides. That includes micro and macro deletions on the Y chromosome, mutations on the X chromosome, mutation on the autosomal genes, mutation on the mitochondrial genome, which are known inherited maternally. And all of these leads to abnormal semen profile. All leads to abnormal semen profile. Three major causes. So there are three major abnormalities: azoospermia, oligozoospermia, teratozoospermia, and asthenozoospermia. And sometimes you get all the combinations of astheno, terato, zoospermia. Yeah, let's look at various causes, particularly the Y chromosome. Look at, there are three azoospermic factors, factor A, B, and C, and there are several genes in those regions, and there are several repetitive sequence in the spanning entire Y chromosome. Specifically, there are several repetitive sequence. One can able to identify the deletion of that using several STS markers. And then we looked at, of course, it's very very long back we did this study. By PCR, you can see there are few deletions in the isoformic factor A, the isoformic factor B, and isoformic factor C. There are lot more, and there are individuals carrying both isoformic factor B and C region, and also the heterochromatic region, which of course we confirmed by southern hybridization. When you come to azoospermic factor C region, there are 4 copies of DAS gene, that's a deleted azoospermic factor gene. DAS 1, 2, 3, and 4. Again, you have lot of repetitive sequence, particularly there are palindromic sequences. And for example, B1 and B3 we combine together and delete this entire region. Similarly, G1, G2 delete the entire region. So we again use several STS markers, try to map those regions, and these are some of the deletions which we have mapped, and of course we confirmed by southern hybridization, where you can see deletion of azoospermic factor dash 1, we have 4, 3, and sometimes there's a combination of those. This is about the Y chromosome, micro deletion plus the gas copy deletion. The X chromosome gene, called androgen receptor gene, is again the mutation of that gene plus the CAGT expansion was found to be associated with the infertility in some other populations, but we did not find that in our population, where you can see the distribution of CAGT in both fertile and infertile males are also the same. But of course, we use this particular gene to understand the sex determination in large number of individuals. We found several individuals with a single base mutation in androgen receptor gene, where they're supposed to be male with the Y chromosome. They're all with the female phenotype. So we're not going to that detail. Then we looked at several autosomal genes, right from genes that are involved in spermiogenesis. In the process of spermiogenesis, where spermatic becomes matured sperm, there are a lot of change happens here. In fact, there are several genes that are involved here. The major change here is the histones are replaced by the proteins, and the DNA is tightly packed. As you see here, the 15% of sperm DNA retained in the nucleosome structure, whereas here, 85% of the sperm is the form of highly compacted nucleoprotein complex. That's the process, and there are several genes involved in that. UD2, the protamine, transition nuclear protein, and so on. So we looked at all of those. So I'm not going to detail, but we looked at there are certain percentage. This slide is different. There's another one, estrogen receptor gene. That's about some of the nuclear genes, and this is about the mitochondrial DNA mutations, where we all know the mitochondrial DNA inherited maternally, again mutation that causes low sperm motility. So now all of this together makes about 1/4 of the genetic cause for male infertility. So that's when we thought that we have more than or approximately 3/4 of the cases where we don't have any answer. We thought that doing exome sequencing would help us in understanding this particular aspect. So we sequence initially 50 non-obstructive azoospermic men who are not having any of those abnormalities, and we duplicated whatever the variants, about 440 variants we have identified from 39 genes in large number of infertile and fertile individuals. 844 infertile individuals, and these are some of the classifications, and 700 plus we identify two different kind of variants. One is the rare variants, which is completely absent in the fertile individuals. Of course, there are a couple of them showed very low frequency, which has been reported elsewhere, but it's not finding anywhere in our sample. Of course, these are found in, as I mentioned. So, of this 10 genes that are here, 5 of them are normal genes, which were not reported anywhere. And the second category, these are associated variants. Okay, you can see some genes are with multiple variants, and 2 genes show very highly significant value, and particularly centrin, and that we thought we should characterize because it's a small gene, and we thought of characterizing that. Before going into that, we wanted to analyze a large number of samples to find out is there any other additional mutation. We sequenced complete gene in 1,000 plus sample and infertile individual and 874 fertile men. And you can see that somewhat variant. This is the one which we have found by exome sequencing, and the 5' UTR variant was found by sequencing particular gene. Then we started characterizing those variants. First, we took the 5' UTR variant, and we have done the luciferase assay, the promoter alone, with the wild type, to the mutant. And you can see the mutant is overexpressed. Sometimes overexpressed also is not good for health. And we found what was the reason for that. Actually, in this particular case, the site where you have done the bisulfite sequencing to understand the any difference in the material site, where you can see this case C are being converted into G, whereas this particular site that we found the mutation remains there, therefore log is not controlling the expression of it. And this protein has not been characterized, therefore we thought we isolate the protein, characterize that by physically. And the good thing is that in CCMP we have large number of groups work on very diverse area. So one of my colleagues, Yogendra Sharma, is a biophysicist, he works on calcium binding protein, and this is centrin is a calcium binding protein. Thought let's characterize that by. We used a different aspect. One is we looked at the second structure with without calcium in wild type with calcium. Similarly, without calcium and with calcium in the mutant. And you can see secondary structure difference in the mutant, whereas the tertiary structure does not do. The ANS fluorescence, as you see here, is the wild type and is a mutant. You can see the shift here, suggesting that the mutant alters not only secondary structure but also other factors. Then we thought of characterizing the mutant in a HEK cell line. This is the wild type, where the centrin is located in the centrosome, and where you see the wild type, this different stage of cell division, where you see 2 dots, suggesting that there's no problem at all here, and this is gamma tubulin, alpha tubulin structure is absolutely fine. It goes into that stage. Whereas in the mutant, where you see that the wild type again moves apart, in the mutant, we started looking at the multiples of the centrosome. 3 dots, 4, and there's an anaphase bridge, the telophase bridge clearly showing. So what all this indicates is because of overexpression, that's one reason. Here because of the mutation, and the cell is not able to divide properly because of multiple central here. Therefore, there is a meiotic arrest, and most of the samples should analyze are azoospermic individual, justifying that this mutation is the ultimate cause. Wanted to tell you about the wild type and the mutant, and this set is the centrosome 14, where you see that here mitosis, and here where you can see in the mutant set multiple central. Once again, you can see the number, the control is the wild type and mutant is wild type. We also looked at the cilia because there are a large number of diseases are associated with cilia leg, called separate group of disease called ciliopathy. In the wild type, as you see here, the cilia leg is smaller compared to the mutant, as you see here, the control, wild type, and the mutant. While it's changing the length of the cilia assembly is intact, microstructure. Where again you can see the multiple centrosomes, and it's not showing the assembly, it's also not showing other shape except the length of the cilia. Of course, all of this put together, and this paper was published very recently and online. It will come in the version. We started looking at the functional aspects of other genes, and text 13 has already been done in the papers and the blue, where we have knocked out text 13b, and where we looked at the proteome of that, and we identified there are several proteins which are involved in glycolysis or upregulated, and other aspects, but mitochondrial proteins are down-regulated. And we correlated that with some of the findings which earlier found that the mitochondria is essential for daily circulation. Of course, I'm not talking about that. Now, I want to conclude that what our study has done. Our study has done given additional genes which are responsible for infertility, and the screening for observed mutation needs to be done prior to the artificial reproductive technique. Through our collaborators, we came to know that there is a micro deletion on the Y chromosome. When you do multiple times of ART, it always fails. That means the mutation, either the autosomes or the Y chromosome microdeletion, always fails the artificial reproductive technique. Therefore, it tells that before wasting a lot of money, because every cycle one has to spend huge amount of money, rather than wasting that, one has to go for testing for either microdeletion or mutation. And we found the mitochondrial link to infertility text 13b because of the amount of time I did not talk about that. And we characterize a lot of genes which are functionally relevant. And it has a lot of social implications, because I want to highlight this. We found the Y chromosome microdeletion. Of course, the Y chromosome, or before that, I want to highlight that unlike other diseases, which is passed from the father to son, because it's mostly the Y chromosome. If the father had the Y chromosome deletion, it's not now being transmitted to the next generation. That's the end of that particular Y chromosome. Only the de novo mutation happens, of course, that is transmitted. Whereas on the other side, the autosome, again, if the father has the autosomal mutation, he will not be transmitting to the next generation. He will be also infertile. But in this case, just to show for the students, consider these two are autosomes. It is a mitochondrial DNA and it is a Y chromosome. So every individual inherits half of the genome. That means 23 chromosomes from father, 23 from the mother. So this chromosome came from the father, and this from the mother. And, and the Y chromosome goes from father to son to grandson, which we heard a lot of talks yesterday. So this has been ruled out. Now the father's nuclear genome autosomes also ruled out. The majority of the cases, unfortunately, we could not look at the father's Y chromosome or mother's father's autosomes or mother's autosome. But mostly we found are heterozygous. That means only it must have come from the mother. But it goes from the mother, and the mother always blamed the daughter-in-law for not having the child. In this scenario, actually, the mother-in-law transmitting the autosomal mutation to son, and she is responsible for infertility. Similarly, the mitochondrial DNA which comes from mother, you are sure so that of mitochondrial DNA which are responsible for low motility. There's no problem with sperm production. They are either azoospermia or oligozoospermia. That means sperm count is less. But as well as zoospermia, motility is not good. So that scenario again, the mother contribute to that. So in this scenario, actually the mother-in-law needs to be blamed for most of the infertility cases, not the daughter-in-law or son-in-law. So with that, I thank all those who have contributed to the study. Thank you. I kept it short because I want to have more time on this.
Thank you very much for such a illuminating and even light-hearted talk. How many of us would be happy to know that mothers-in-law are to be blamed for something? Yeah, but my question was regarding content one and that mutation. If it is a centrosomal mutation which leads to aneuploidy, what are the chances that these people will also have a predisposition to cancers of some kind? Because they should, and that why should it happen only in males?
Yeah, this was a question we also asked ourselves, but we did not have any data to show that not only cancer, they have any other diseases. But of course, we did not go back to those individuals to find out because they never come back again once they know that they have the factor causing infertility.
No.
But
I mean, if you look at their somatic cells, they also should show aneuploidy.
yeah,
So what's the problem? I mean, at least we could see in their somatic, and if they show aneuploidy in somatic cells, then there is a very high chance. At least they could be warned that they should take care of it.
yeah, probably it's not going into somatic, otherwise a lot more abnormalities. We also did not check in the germ cell because the germ cells are not available for us. This is the,
But even if you look at lymphocytes, I mean, this is just a suggestion because it's a very alarming kind of a situation that we see from this gene and this mutation. So I would imagine that it would be worthwhile looking at least some somatic cells and see whether there is a predisposition.
that's what we wanted. Unfortunately, we can't get their cells.
Hello, this is really a good lecture and thought-provoking. So definitely there will be the question since it's the infertility problems and societal benefit. And I feel that everybody is interested and provocated to have some kind of questions to Professor Thangaraj because it's always amicable presentations, whatever, in so easy way. So I feel there must be some other questions. So due to the constraint of time, I feel that we can have just only one question from here, and he will be available, I feel, for the coming 2 days. So it will be best.
Congratulations. And you know, when I think of Thangaraj, he is situated in a situation where he works on ancient lineage, and evolutionary genetics, and now disease-specific molecular understanding. What I am going to ask you is that in terms of translational therapy, therapeutic intervention, are you going to start a company or already you have some ideas?
No, I don't have any plan to do that. We can take the idea from here.
So it is really fantastic to listen you, sir, for any time. So there will be a lot of time that you will be there, and
Sir, for this rather enlightening talk. And now I request Professor Bandyopadhyay to please present Professor Thangaraj, Dr. Thangaraj with a memento token of our respect and love.
my greatest pleasure, sir.